Good morning students. Welcoming to the next video class children. Have you learned the previous class portion? Say yes or no? Yes. So all are prepared, isn't it? Shall I ask some questions? Yes. Yes or no? Yes. Good. Do you remember the portions for that previous class? Yes. Good. What's the things learned, children? Very good. Multiplication of phages. That is a life cycle of phages. Clear? So the life cycles, they comes under in two types. What are the two major types, children? Lytic cycle and lysogenic cycle. Clear? Lytic cycle and lysogenic cycle. Which cycle comes under a virulent cycle? Very good. Lysogenic cycle is the air virulent cycle, isn't it? So lytic cycle is the virulent cycle. What do you mean by lytic cycle, children? What do you mean by lytic cycle? Very good. Disintegration of this phage DNA to the bacterial cell, isn't it? Disintegration of this, very good, phage DNAs to the bacterial chromosomes. So that is the type is called lytic cycle, isn't it? So this type, so there is the, very good. So that's a phage, that's the young ones, that's the virions are releasing. This is the type is called lytic cycle, isn't it? If that coming to this is the phage DNA particle inside, that is the bacterial chromosome, but not integrated. Integrated means joining of this bacterial chromosome, but here there is no integrations. That is the lytic cycle, the first cycle. Clear, children? Yes, good. The next one, what is that? Very good. So there comes under in 5 steps, isn't it? Do you remember that? Can you able to say? Yes, very good, children. Tell me. Adsorption, penetration, good. Synthesis, assembling or maturation. The final step is release, isn't it? So there we had learned some points, isn't it? First one, adsorption. There we covered, that's a 2 questions. What are the 2 questions, children? Very good. First one is landing and another one is pinning, isn't it? What do you mean by landing, children? Very good. The phage contacting of this two, I mean the phage tail part and the bacterial cell, they make it in that contactions. Okay, I mean, so there only they searching of this, very good, the receptor site, okay, the recognition of the site, they find outing of this site. But the tail fibers, what saccharides molecule is there? Lipopolysaccharides molecule is there. So that is act as a very good receptor site of this phage particles. Clear, children? Say yes or no? Yes, very good, children. Then, so these contaction of this, so there is recognition of this receptor site. Okay, the recognition of this site of this phage to the bacterium, that's known as landing. Very good. What do you mean by pinning, children? Very good. After the contractions, isn't it? So there is a cell wall and phage particles. They are attaching, that's a host bacterial cell, isn't it? So this is the regions, isn't it? Then what will happen? Good. The phage particles, very good, bend it and pin the basal plate to the cell surface. Clear, children? Pin to the, very good, that's a basal plate, pin to the cell surface, fixed to this is the basal plate to the cell wall of this host. I mean host means who's that host? Bacterial cell. Clear, children? Say yes or no? That's known as pinning. Very good, children. Clear? So that is the 2 concept. The next one, penetration. What does that mean by penetration? Entering, that is the phage DNA particle inside to the bacterial cell. Okay, that's known as, very good, penetration. So what do you mean by transfection, children? What do you mean by transfection? Very good. So here the contraction of these two, after the contraction, the cell wall and the basal plate, isn't it? That's the link is there. So here the basal plate is the shortened, and then so just opening that region. Which region? Basal plate. Through that way, DNA alone, the phage DNA, clear children? The phage DNA alone, this is the phage DNA. This is the phage DNA alone enter inside of the very good bacterial chromosomes. That is known as only, that's a linear form of this phage DNA particles alone enter inside the bacterial chromosomes. No? That process is known as that alone, DNA alone injected into the bacterial chromosome. That's known as transfixion. Clear children? The next one. What do you mean by ghost down? Very good. So if that injection of this phage DNA particle remaining only the empty items only there, isn't it? The empty protein code. So this is the empty protein code. So already injected to the DNA to the host cell, isn't it? Then remaining these part alone from living in the outside of the cell, isn't it? So this is a cell living outside of the cell. That's known as, very good. That's another one of the very important 2-mark questions, children. Ghost. Understood, ma'am? Say yes or no? Yes. The next one, third one, synthesis. Synthesis means the DNA particles of the phages entered into the bacterial cell and the host cell. All the machinery items, metabolic activities takes over the control of this, which DNA? Very good, phage DNA. Clear? Then immediately suppress all, otherwise, very good, destroying all the activities of machinery activities of bacterial chromosomes related. Then synthesis of bacterial DNA and then bacterial protein synthesis. Everything is destroyed, and then only the synthesis for that phage DNA related. Understood? Say yes or no? That's the process is called synthesis. Clear? Yes. Next one. What is that next one? Good. Very good. Tell me. Try to tell. Assembly and maturation. Isn't it? Assembling means already that's a previous step. That's a synthesis step. There the parts are very good. So synthesized separately. Isn't it? So that's the individual part, the separate parts is only so here present, isn't it? So there is a protein coat, there is a tubular and a tail fiber. This line, that's a separate parts is here present. Clear, children? So these parts are this is the steps. The synthesis step is they separated in a separate parts wise. Okay. So here this is the separated part. The assembling process is going on in that assembly and maturation steps. Clear, ma'am? What do you mean by maturation, children? Assembling of this phage particle is known as maturation. Okay, the assembling of the phage particle is known as maturations. Clear, children? Yes. Next one. How many minutes the phage particles they are releasing around, that's a 300 numbers. Very good. 20 minutes time. Okay, after that 20 minutes time, how many phages are maturated? Very good. 200 phages particles are maturated. Clear, children? Yes. Then final step is releasing. Releasing means so the occupation, the accommodation of this more number of this phage particle inside this is a host cell. Host cell means bacterial cell. Then what will happen? Not have that is a space, the more accommodation means, isn't it? So that time, so the breaking out. Breaking means lysis of this is the host cell, they coming out of this. That's the new phage particles. Clear, children? Understood, no? The new phage particles are that new virions are coming out. Clear? This is the first step. So this is the first multiplication of the phage, or that's the first life cycle of the phage is a lytic cycle. Clear? The next one is lysogenic cycle. What's mean by lysogenic cycle, children? What does mean by lysogenic cycle? Can you able to say? Very good. Lysogenic cycle means, so here that's a phage DNAs Very good. So entered in that cell, isn't it? The host cell. At that time, what will happen? This is the phage DNA. Very good. Integrated with the which one? Bacterial chromosomes. Clear, children? So the integrated form of this phage DNA. So that is the multiplication phages, or that is the life cycle of this phages. That's the type is we are calling as a lysogenic cycle. Clear? Then what do you mean by propage, children? Very, very important, one of the 3-mark question. What do you mean by propage? Very good. So as soon as the phage particles, first initial stages, they coming out in a linear form. Okay? Immediately comes to, this is the area. I mean, where is the area? Inside the bacterial chromosome. So they make it in form, the linear form of this very good phage DNA particle, they make it in a circular form. Clear? Then through recombination method, okay, by that recombination methods only, this is the bacterial chromosome and the phage DNAs are integrated. Okay, the integrated form of this phage DNA is known as propage. Clear, children? Say yes or no? The integrated form of the phage DNA is known as propage. Clear, children? Say yes or no? So then what will happen? The phage only, very good, that's all the phage activities they controlled by, that is the 2 receptor proteins, isn't it? That receptor protein acts as that is the phage genes. Clear? So then what will happen? Suppose if this is the multiplication of this bacterial cells means, so along with the propage only, okay, already you know this is the propage, integrated form of the phage DNA. If we want to multiplying, or that's if we want to dividing this bacterial cell means along with the propage of this phage DNA. Okay, so that's a propage stages only that can be divided. Understood, no? Say yes or no? So this is the second cycles of this lysogenic cycle, or that is the lysogenic of this phages. Clear, pa? Say yes or no? Yes, good, very good. Then the next one we are going to learn about, this is the viral diseases. Okay, so the next one we are moving to learn about, this is the viral diseases. Before this viral diseases, we want to see some of the terms of this virus. Clear? So here, this virus, they are mentioning in 4 terms. First one is viral, viroid, virusoid and virions. So many 2-mark and 1-word questions are there, children. So you must thoroughly prepare this one. Okay, first all of you listen carefully, and after that you must prepare these all the 4 2-mark, and then also in between there some 1-word questions also there. Okay, so all of you listen carefully. First one, what do you mean by virion? Virion means infectious virus particle is known as, okay, the infectious virus particle is called virion. So this cannot be multiplying outside the host cell, okay, not replicating of this outside of the host organism. So that is the definition. So that is the infectious viruses particle. So this one not replicating outside the host organism or host cell. Clear, children? Next one, what do you mean by viroid? Okay, this viroid means, so this is the one of the circular molecule. Okay, so this is one of the circular molecule. This is a single stranded RNA is present, but that is the without that is the protein coat. Clear, children? So this is the one of the singular, very good, circular single stranded RNA molecule, but without that is the protein coat. You know that what do you mean by core substance and what do you mean by protein coat? So here this type of this virion, okay, so this is the virion, not have, that is the absence of this protein coat. Clear, children? So this term that's discovered by that person name is known as Theo. Okay, so that's the person they discovered, that's viral, that's the person name is Teodynia in the year of 1971. These are all the very important word based questions. Okay children, so don't forget. Then, so this viroid, the causing of this, some of the diseases in plant side. Okay, because this virus is infecting that's a normal plant, so they cause some of the diseases. So that's the host of this plant is citrus and potato. Okay, citrus, that is one of the disease name is exocotis. Okay, the next one is potato. So there they causing in that one of the diseases is the spindle. Okay, so there is the spindle tubers. So that is the causing of this one of the host plant is the potato. Understood, ma? Yes. Next one we are moving this next one of this term, virusoid. Okay. What do you mean by virusoid? What do you mean by virusoid means? So this is one of the very small circular form of the RNA molecule. But this one is similar to this is a viroid. Okay. So this is similar to that's a viroid. But anyway, these virusoids are mostly there is their joining with that is the viroid molecule. Okay, the virus's particles only they presence in that is the virusoids. Clear children? Say yes or no? Yes. So there is a very small circular particles of the RNA particles. So this one is they presence in a complex structures of the virus particles. Okay, this similar if we are looking means so that is the similar to that is the previous one, that is a viroid in nature. Understood, ma'am? Say yes or no? Yes. Next one. So here this term is they discovered that a person name is known as J.W. Randless and co-workers. Okay. So that's the Randless and co-workers in the year of 1981. Okay. In the year of they discovered that's year of 1981. Okay. So many one words are there. Don't forget. So all should remember in your mind. Next one, virions. Virions means, so this is the one of the infectious proteinaceous particles. The protein related one of the infectious particle is known as virions. Clear? So this virions are discovered, that's the person name is known as Stanley B. Brusman. Okay? Stanley B. Brusman in the year of 1982. Okay? So these virions are the first one they are fighting in that is that's a fatal degenerative disorders and also affecting in its a central nervous system. They affecting of this the human beings and also animal sides. This is the virions are affecting and cause some of the diseases. Okay, that related they are mentioning in a two examples of the diseases in your syllabus children. So that is first one as Creutzfeldt-Jakob disease and another one as bovine spongiform encephalopathy. Okay, so these are the two diseases. They are commonly called as one as mad cow disease and another one as scurvy disease of sheep. Okay, so these are the two diseases we are commonly, these two diseases we are called locally, this is the name of the diseases. Okay, the first one, so this one is they affecting in the fatal degenerative disorders and also they affecting in a central nervous system. Okay, so that is affected in that it's a host of in the animal side and also that is the human beings. Understood children? Say yes or no? Yes. Next one, we are moving in the some of the viral diseases in that animal side, plant side and human beings. Okay, if suppose one normal plant is there means, so that virus infecting or affected that normals means, so that one they get it in a or they affecting in a certain diseases. So these regarding the viral diseases or they comes to that plant side and animal side and then human beings. Okay, then just we are going to learn about that. So what are the diseases or comes in the each host organisms? I mean plant side, animal side and human beings. Clear children? Yes. Let us learn that one by one. First one, the plant diseases of this viral diseases or they comes under in that's tobacco, tomato and then cucumber, sugarcane and then cauliflower. So these all the host plants, clear? These all the host plants, they appear in that's a mosaic-like symptoms. This is the disease name is we are called as the name is known as mosaic diseases. Mosaic diseases means already I taught this one. So there is a yellow and greenish coloring of the dot-like appearance. So there is presence in the middle of this regions, isn't it? So that is the region they appear for this green and that's a yellowish color. The dot-like representation is there. Just they looks like the mosaic-like symptoms. Understood, ma? What are the plants? Very good. Cauliflower, tomato, tobacco, very good. Cucumber and very good. The final sugarcane, isn't it? So these all the plants, so they affecting in that is what is the type of the viral disease name? Mosaic diseases. Clear? The next one is leaf roll, or that is the leaf curling. Okay, the curling of the leaf. Suppose if that virus is affected in that particular plant, so that's the leaves or the symptoms they appearing in the leaves, the curls, or that's the leaves are rotating. That is, that's two examples of the host plants. So first one is potato and that is the papaya. Okay, so these two plants, the viral diseases they affected, that's one of the normal plant. The viral diseases they appearing in a leaf rolling or that's a leaf curling. Clear? The next one of the diseases is the Thungro diseases. Thungro diseases means so that and also just like a grasshopper like. So that is the things only, they cutting of this, they touching of this is a particular rice plants. Rice plants means paddy plant. So that things, so that's a very good spreading of this viral diseases. Clear? That is a Thungro disease of rice. That's a disease name is known as the viral diseases name is known as Thungro diseases. That's a host organism is the host plant is the paddy plant or rice. Clear, children? Yes. Next one, there is a bungee top of banana. Bungee top of banana means, so if you are looking in that is a cultivatable land of this banana field means, sometimes the banana plants are not properly producing in that is the flowers, isn't it? The flowers and fruits. I mean the topmost region, so there never produces in that flowers, children, but that is the very tufted, I mean very cleft, or that is the very crowded form of the leaves only they present in the topmost regions of the banana plant. Clear? That is the diseases, the symptoms is they appearing in that is the banana plant. That disease's name is we are called as the name is known as bungee top of banana. Understood, children? Say yes or no? So these are the most of the diseases they comes to that is the viral diseases in plant soil. Clear? Say yes or no? Yes. Next one, that is the animal diseases. Animal diseases, so they are giving in a foot and mouth diseases of cattle. That's one of the diseases, and foot and mouth regions only they present in that is the diseases. Clear? Next one, rabies diseases. Rabies, you know that. Nai kidding, very nai, that's a lie. Okay. Then encephalomyelitis of cause. Encephalomyelitis means, so this diseases comes to that animals means, so they linking to that is the very good. So the brain to backbone. Okay, the brain to backbone they linking and affected that's a particular portions. Understood, ma? Yes. Next one, we are moving to that is the human diseases. Human diseases, the viral related diseases. Will you able to say this is some of the diseases for that viral diseases in human beings? Can you able to say? Yes, tell me. Do you know? Yes, good. Tell me. Common cold? Yes, good. That is one of the viral diseases. Yes, the next. Chicken pox, chikungunya. Isn't it? The next one, SARS, 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 AIDS, yes, good. Then measles, polio, yes, good. Then mumps, rabies, cancer, chickenpox, smallpox. So these all the diseases are they related to comes to the host of this human beings. Okay. So here this is the viral diseases here comes to that human beings. Understood, children? So these are the some of the common diseases, the viral diseases they comes to that plant side, animal side and human diseases. Understood, ma'am? Yes. Then totally I am once again go through that first one. The four terms viroid, virucide, virions and perions. Okay, so these related, these four terms, what is that, what is that, just a one word like questions, what is that, what is that. Then after that, apart from that, you should prepare that is the mainly one word question. In which year, which person discovered, what's the diseases is coming related to that, these are the things. Understood, ma? The next one, you must prepare in some of the viral diseases. The diseases I had explained detailedly. So you must just prepare that is the what are the diseases affecting in a plant side, animal side and then human being side. Okay. So all should prepare some of this 2 mark and then one words based, and also then finally the viral diseases, sometimes they will ask, this is the full tabular columns means that's a 5 mark based, otherwise they will ask that is a some of the 2 or 3 mark based. Write names some of this viral diseases in plants, or they will ask that is a mention some of the diseases, viral diseases in human beings and mention some of the viral diseases in animals. Okay, so this like only they will frame in your board examination, etc. So all should prepare thoroughly. Thank you, students. Thank you for watching this video.